Hi everyone, my name is Delaram. I'm a PhD student in Gary Bader's lab at the University of Toronto, and today I'll be talking about a project on interpretable factor decomposition of single-cell transcriptomics map of the rat liver. Single-cell RNA-seq technologies have equipped biologists with a powerful tool to profile heterogeneous cellular systems such as the liver. Improvements in cost, throughput, and accessibility of such technologies have led to the fast growth of single-cell genomics data. However, many technical and biological factors make the integrative analysis of single-cell data challenging. In this project, we aim to use the single-cell RNA-seq technology to understand the cellular landscape of the healthy rat liver. Liver single-cell maps are known to be affected by a sample-specific batch effect caused by contamination of mRNA from sensitive cells, like hepatocytes, that are damaged during the cell dissociation process and leak their cytoplasmic content into the media. The mRNA molecules of the lyse cells, called ambient RNA, confound the data and cause issues in the downstream analysis. For instance, pathway enrichment analysis applied to the differentially expressed genes failed to work in our data, and the cluster annotation and string comparison were inconclusive. These challenges highlight a need for a systematic approach to dissect the biological and technical signals of the data from each other. We can categorize sources of variation in single-cell RNA-seq maps into two classes of technical and biological. Technical confounders can be introduced at every experimental step, from cell dissociation procedure to sequencing, and some examples of biological variability are genetic background, sex, age, or the strain variations between the samples. In the ideal case, we would like to remove the technical confounders while preserving the biological variations of our interest as much as possible. Matrix factorization is a group of well-established analytical methods in statistics and machine learning used to describe variability among measured variables in terms of a lower number of unobserved variables, or factors. These methods decompose the gene expression matrix into two related matrices. The first one is the loading matrix, which defines the relationship between the genes and the factors and can be used for pathway analysis and biomarker discovery. And the second one is the score matrix, which represents a relationship between the factors and the cells and is usually utilized for cluster analysis and visualization. In many cases, the inferred dimensions from standard matrix factorization methods may not align with biologically meaningful gene expression programs, and nonlinear methods often lack interpretability. One of the most prominent factorization methods is principal component analysis, or PCA, which identifies orthogonal dimensions that capture the maximum amount of variation in the data. Maximizing the variability captured in specific factors can lead to the conflation of signals from multiple biological processes in a single component, which makes the interpretation of the learned factors more complicated. One approach for improving the interpretability of the extracted PCs is to apply rotations, such as varimax, to the PCA loading matrix. This higher interpretability is mathematically achieved by maximizing the variance of the squared loadings in each factor, leading to factors which are constituted by only a few highly loaded variables, while the remaining variables have near zero loadings. Let's look at an example of varimax in more detail. I'm visualizing the distribution of cells over PCs before and after rotation. On the left side, we have the PC number 2 on the y-axis and PC number 1 on the x-axis. And as you can see, the signal is a lot more clear after applying the rotation. And we can associate varimax PC number 1 with the hepatocytes and varimax PC number 2 with the macrophage population. To automate the interpretation of the varimax factors, we use the combination of correlation analysis and random forest binary classifiers. The correlation between the average gene expression of each cell cluster and the loading score of each varimax PC was used to match factors with cell type identity programs. An example of a factor and cellular identity match has been demonstrated on the right. Once a cluster and a factor have been matched, we evaluate the biological function of that cluster by performing pathway analysis on the loadings of the corresponding factor. A random forest model was used to identify the varimax factors that capture strain-specific variations. This classifier was trained to predict the strain attributes of each cell by using Varimax factors as input features. The factor with the highest feature importance score was chosen as the best match factor for the predicted covariate. Using this approach, two main strain-specific factors were identified which represent variation within the hepatocyte and macrophage populations. The top two hits were Varimax factors number 5 and number 15. Both factors indicated near zero correlation with the QC covariates, suggesting that they have captured biological rather than technical variations. Interpretation of the macrophage-related factor using pathway analysis and transcription factor gene set enrichment analysis highlighted the enrichment of pro-inflammatory signals in the Lewis-rat myeloid cells, suggesting that the baseline hepatic microenvironment in the Lewis rat is more pro-inflammatory compared to the Darko-Goody strain. To functionally validate the computationally inferred strain-specific differences, we performed ex vivo LPS stimulation followed by intracellular cytokine staining to measure myeloid cell inflammatory response. We found that Lewis intrahepatic myeloid cells secreted overall more TNF-alpha in response to LPS stimulation compared to Darko-Goody, suggesting that hepatic myeloid cells may play a role in mediating differential response to liver transplants among these rat strains. Taken together, we present a novel pipeline based on Varimax PCA for the identification of biological and technical sources of variation within the data and indicate its utility for uncovering key insights into strain-specific differences in the first single-cell transcriptomics map of the healthy rat liver. And with that, I would like to thank you all for listening, and please make sure to check out my poster if you'd like to learn more.